Okay. What are you doing in terms of, are you ready to listen? Okay, this was, I know it was hard. So I know that everyone is hating me now horribly because I took you so much time. But believe me, this review will be very useful when you get quiz. Then you will be happy. Okay. Mm. I don't think we will have time to finish review for unit 2, but I think you're sick of metabolic diversity for like at least 1 week, right? Yeah. So leave me alone with electrons, please. Anyway, so that's why I will put you back a little bit on the first week when we talked about evolution generally. Why this matters? Because these are main concepts. What that means is that the review session contains main concepts that I want you to know. Mostly, so that doesn't mean you should not study lectures in general. You should, because there will be questions. But in terms of organisms that you need to memorize, because there will be many Latin names, I use these review sessions to give you like hint what I want you to know, which organism. Because God help me, nobody can memorize all of that. And I'm not going to ask it. So I give you a hint. But when it comes to the midterm will be, and I decided that recently, midterm will be less detailed than quizzes because you will have more to learn. So quiz will contain only 2 units, so I will ask more details, but midterm will be more general concepts. So, and I'm going to probably give you, not probably, I'm going to give you a review session. If I'm not here, Brittany will. So it's going to be okay. So, going back to the formation of early Earth and living fossils, which is something that am I recording? Yes, I do record myself. Okay. So certain numbers that I want you to know, even though you know how much I do not like Earth, but you do need to know that Earth is formed 4.5 billion years ago. And this abbreviation BYA means billion years ago. So what is important to know is that Earth was completely inhospitable environment, which means not good for us because we were not existing at the time. It was hot, there was no oxygen, water was present only as a vapor. And you need to know those 4, Hadean, Archean, Phanerozoic, and Proterozoic. And at the end of Hadean, cellular life showed up. That was 4 billion years ago. So first was Earth and then was cellular life. The way we always think, how we say that life developed, there are 3 theories, and one of them is completely incorrect, probably, but the other 2 are kind of intercalating with each other. So the first surface origin is something that we talked a lot, well, not talk, we just mentioned, and to me this made no sense, but again, I will ask theory. Basically, what surface theory says is that there was a primordial soup, primordial organic inorganic soup, and the one formed as a spontaneous arisal of membrane enclosed structure from that soup of organic and inorganic matter. So again, for me, that really didn't really, I didn't buy that story. But subsurface origin, it makes a lot of sense, at least to me. So subsurface origin hypothesis is is related to the interaction between. So it basically says that life started beneath the ocean, and there was an interaction between hot water, like from beneath the surface of the ocean, like alkaline hot water, and the cold acidic ocean water. Because beneath the ocean surface, there were a lot of hydrogen sulfides and hydrogen. So they were present as a constant energy supply because those are all highly reduced compounds. So on the surface, there was an iron. So you have like hydrogen sulfide, hydrogen iron that are interacting beneath the surface, on the surface. So interaction between sulfides and iron literally led to the formation of metal precipitates. And those metal precipitates were actually preceding the enzymes because they were catalyzing formation of first molecules like amino acids, sugars, and bases, which was a base for development of first macromolecules. So iron sulfides as catalyzers were part of subsurface hypothesis. The one that is highly accepted is RNA world hypothesis, which says that RNA molecule was first entrapped. So it was entrapped by self-replicating organisms. So the first molecules were self-replicating RNAs. So RNA was the first catalyzer, the one that catalyzed the formation of simple peptides. And those peptides were coated with minerals. Again, this is very similar to metal sulfide. So who was the one that actually catalyzed it? So one hypothesis says that RNA was the one that catalyzed it. And that's now broadly accepted. So according to the RNA world theory, the order of events was very simple, and it started with formation of amino acid nucleotides and sugar, which was created by RNA catalytic world. So self-replicated RNA was the first molecule that catalyzed formation of everything else. And then as RNA was catalyzing amino acids, sugars, and bases, it also kept forming complex molecules. So RNA catalysis was very active. And then when you had all those complex molecules like complex peptides, complex sugars, then RNA was not really able to be maintained because it was highly unstable. It was single-stranded. So DNA replaced RNA. So when DNA replaced RNA, the next step is lipid arousal, and you have to have lipid arousal because that's what creates a membrane, like a surrounding a compartment. So when lipid arose, so you had RNA, you had DNA, and you had lipids. Lipids surrounded RNA and DNA, and that's the moment when last universal common ancestor, or LUCA, appeared. And when LUCA appeared, that was the first cellular life, and LUCA diverged to bacteria and archaea. That's, again, the order of events. So bacteria and archaea branched from LUCA, and early in their structure, you had lipids. So they were one of the early diversities that we see between bacteria and archaea is that difference in ester versus ether bond, because in archaea you will have ether bond between glycerol moiety and isoprenes, and in bacteria you will have ester bonds between fatty acids and glycerol molecules. So that's actually the indicator of evolutionary phenomenon. Both lineages have specific metabolism, so they are different, but the first metabolism that we think that developed was methanogenesis in archaea almost 4 billion years ago, 3.9, but I would accept them because it's not really easy to determine. But it's important that we think now that was the first one. And next one was anoxygenic photosynthesis, which were green and purple bacteria that consumed hydrogen sulfide. Alongside hydrogen sulfide consuming purple and green photosynthetic bacteria, you will have development of hydrogen oxidating organisms. And that all happened in between of, like, after LUCA diverged, like 3.9 billion years ago, all the way down to the 2.8 billion years ago when cyanobacteria showed up. So that was the end of second stage, which was Archean. So you had Hadea and then you had Archean. And the Archean was ended by big oxygenation event. So cyanobacteria appeared 2.8 billion years ago. But remember, the big oxygenation event did not appear a few billion years after that. And here is the reason why. And these things I will really ask. It's important to understand. So even though cyanobacteria produced oxygen reacted with all available iron on early Earth, and it made these insoluble iron banded formations. So oxygen was entrapped. So iron in those banded iron formation was ferric iron. So when it got oxidized by oxygen, it became insoluble. And that is what actually that's what entrapped oxygen, and nothing could develop. But when all iron was exhausted, the oxygen level made by cyanobacteria would increase, and when Earth became anoxic, that actually happened 2 billion years ago. So it happened 2.8 billion years ago when cyanobacteria developed, all the way down to the 2 billion years ago when actually there was an oxygenation event, and that oxygen arose, like it accumulated, and you could get eukaryotic cell to develop. So another reason why eukaryotes really required a lot of oxygen is because you remember how Earth was in a hospitable environment, so there was really high UV radiation, and in order to protect Earth from UV radiation, you have to have ozone, and ozone is O3 molecule, and to form O3 molecule, you need to have oxygen. So even though cyanobacteria made it because iron was available, that entrapped oxygen, so UV was still very active, and eukaryotes could not develop on the Earth's surface. And in terms of eukaryotic cell development, we have two theories, actually hypotheses 1 and 2. So in hypothesis 1, which is more simple, DNA was accumulated, so DNA simple got accumulated after it replaced the RNA, and that simple DNA accumulation led to the formation of nucleus. So the first cell initially had nucleus, and the next so after nucleus, like cell was formed with a nucleus, that cell now ingested chloroplast and mitochondria, and then you got the first cell. Hypothesis 2, which is probably more accurate, again, it's only opinion based, that first early cell was actually archaea, so that was the host that consumed oxygen, and to ensure energy supply, that archaea, oxygen-consuming archaea, ingested hydrogen producing bacteria because it is energy source. And nowadays you actually see hydrogenosomes organelles in certain organisms, which means that this theory probably is accurate. So so eukaryotic cell is nothing else but chimera, which is the mix between archaeal and bacterial cell. So that's how we call it. It's a mixture. They have eukaryotes have a lipids that are same like those found in bacteria, so glycerol moiety fatty acids. Archaea have completely different lipids, but eukaryotes surprisingly have transcription and translation material that more resembles archaean than bacteria. So what endosymbiotic theory says is that chemorganotropic bacterium host, now we know it's actually archaeum, so take that with a grain of salt, be careful, we will say it's archaea. So this host injected oxygen consuming and ATP producing mitochondria, and eventually this host injects chloroplast where oxygen was made. So who was the host? That's usually the question that I was getting. I believe that new theories are agreeing that the host was archaea, and that archaea injected hydrogen producing organisms and needed that for energy production. Again, don't worry about that. I will never ask something that is so complicated, and I will not take the time for that. What I will ask is those evidences that support endosymbiotic theory are very important. Like this will be something that comes into the story all the time. I want you to know this for every day, night, every class, every time when you finish this class. And whenever I see you on campus, I will ask you this for. And if you don't know, I'm not going to buy you coffee. But if you do, I will. I promise. Really. I'm saying that because I know everyone will forget. So anyway, so what are evidences of endosymbiotic theory is how we know that there was a host that ingested chloroplast and mitochondria, how we know that the eukaryotic cell is chimera, because when we look at mitochondria and chloroplasts, we will see that they have ribosomes, which are 70S, which is actually similar, like not similar, they're like prokaryotic ribosomes. And they will have rRNA, so they will have its own ribosomes inside. So you have cytoplasmic ribosomes, you have mitochondrial and chloroplast ribosomes, and they will be 16S rRNA. They will have 16S rRNA, And same antibiotics, surprisingly, that affect ribosomal functions in bacteria will inhibit ribosomes in mitochondria and chloroplasts. They're the same thing. There is independent DNA in mitochondria and chloroplasts, and it's a covalently circular form, like in bacteria. And many signs of bacteria are present in eukaryotic organelles. One of the signs are those hydrogenosomes. So they are one of the signs. So do we know that this is primary? And this table is something that I actually quite insist on. What are the differences, so characteristics of bacteria, archaea, and eukaryotes? Look this table carefully. So if this is not enough for you, I don't know what else is. So this table is very important. Evidences of early microbial life are stromatolids. So they're ancient stromatolids, like we call them microbial fossils, even though they're not like a real fossil, they're not dinos. The early ancient stromatolites are formed by phototrophic bacteria, while some younger modern stromatolites are formed by phototrophic oxygen-evolving bacteria. What we use to determine evolutionary history of life, we use DNA and rRNA. So DNA is a record of past evolutionary events, and it's used to determine phylogeny, which is evolutionary history of organism. Tree of life will be more clear, like molecular phylogeny and tree of life will be more clear later in the semester. For now, know only that ribosomal rRNA was molecule that revolutionized understanding of microbes. And we didn't really know a lot about evolutionary history of microbes before until we figured out that rRNA or 16S rRNA can be used to make those trees of life. There are few old trees of life, and obviously Haeckel's one is not used because at the time when Haeckel developed it, he didn't really understand how life is formed. But in this Haeckel tree, Monera is on the bottom of phylogenic tree, and Monera was actually the ancestor of all products, and the Monera branched into the three branches: protists, algae, protists, plants, and animals. To add up on the Haeckel's tree, there was this Whittaker, so that Monera was the primary life on the bottom of phylogenetic tree, and Monera was ancestor of protists, but unlike Haeckel, he recognized that fungi are separate branches. So you'll see the branches from protists, you'll see fungi, plants, and animals. And the last one was Woos, who actually used rRNA to define evolutionary relationship between organisms. Why we think that RNA is the best molecule to determine phylogeny, and it is. So again, this is same like those four evidences of endosymbiotic theory. The reasons why we use rRNA is something that I will always ask. And the reason why rRNAs are used to determine evolutionary history of organisms is because first, they are universally distributed. They are very constant functionally. They always do the same thing. They're constitutes of ribosomes, very conserved, they're slowly changing, and they have a proper length like long enough to help determine evolutionary relationship. I think they're like 1500 base pair. So the modern tree of life, which Brittany will tell you more about when seeing the product, is based on 16 RNA tree. And on the bottom of this tree of life is the last universal common ancestor, or LUCA. We say that one of the sources of diversity was horizontal gene transfer, or AGT, which in early history of life was very intensive, but eventually it decreased as that barrier between branches was more strong. So that horizontal gene transfer decreased when bacteria and archaea split from LUCA, and eukaryotes split from archaea 2.8 billion years ago after ozone shield was developed. What was the next? Again, phylogenetic tree, how it looks. It will be more clear when Brittany actually teaches you how to create it, but these are just some structural components like nodes and branches. You have pictures that I forgot to put here. Know only that nodes are the showing the stage of evolution where ancestors diverged in two lines, and branch length indicates the number of changes, so longer branch, more changes. Two types of phylogenetic tree, rooted and non-rooted. Most of the time you see a rooted tree, and in rooted tree you actually see position of ancestors for all organisms. Unrooted tree, I don't remember when I saw it last time, and said that this actually it only shows relative history, but it doesn't show common ancestors. And I'm thinking that, well, I have 9 minutes. How we use DNA sequences in phylogenetic analysis of life? Well, we extract DNA and we sequence genome directly. We amplify specific gene via PCR, and then we can run that amplified PCR on gel, extract it in sequence, and see which gene is present there. In terms of microbial evolution, we define evolution as the change in frequencies of alleles in a set of organisms over time. So alleles are just alternative forms of different genes, and the way how new alleles arise are mutations and recombinations. So you have to know what are sources of genetic diversity, which means you have to understand mutations and recombinations. Which brings me to the next slide. What are mutations? Random changes in DNA sequence accumulate over time, and they are fundamental source of diversity, and they can be neutral, which means they don't cause any changes in adaptation. They can be beneficial, which means they will give cells higher level of adaptation and increase fitness, or they can be detrimental, which means they can cause the death of the organism or less adaptation. The way mutations can happen in several forms. It can be substitution, which means the replacement of a certain bridging DNA, or deletion, which means removing a certain nucleotide or portion of a gene, or insertion and duplication, which means adding a certain molecule, adding a certain portion of DNA to that. Recombinations are another source of diversity and can be homolog and non-homolog. Homolog recombination requires a different set of DNA sequences that surrounds a gene that will be recombined. Non-homolog does not require specific sequence, and it's less common. So the source of diversity are mutations and recombinations, but mechanisms of evolution that actually determine which one will stay, which one will go, are selection and genetic drift. So again, mutations and recombination cause variations, while selection and genetic drift will decide what will stay, what will go. So in terms of selection, so that's basically the ability of organism to adapt and produce progeny in certain conditions, while genetic drift is just a random process that can cause changes in the gene frequencies, and genetic drift is very simple related to pathogens. And we have here new traits that can evolve quickly by microorganisms, but because we have like 10 more slides and I don't want to rush through them, I will stop here and we will continue doing, Brittany, just make a note. Yeah. So we will continue next time when we have a class, and I will again record the lecture. So we will talk about examples of Rhodobacter and E. coli.